Okay, hi everyone, and welcome back to the main session. I'm Tuomas Kilpeläinen, one of the group leaders at the Center for Basic Metabolic Research, and will be chairing this session. And the talk for this session will be a virtual talk because the speaker could unfortunately not be here with us in person. But even so, it's a great pleasure to introduce Professor Karen Mohlke from the University of North Carolina in Chapel Hill, where Karen is also Oliver Smithies Investigator and an Associate Chair of Research in the Department of Genetics. And Karen has been for many, many years been a leader in the human genomics field what comes to the discovery of genetic risk variants for metabolic diseases in terms of connecting the genetic variants to gene functional changes, particularly the levels of gene expression and understanding how the function of genes is regulated in the tissues that are most relevant for metabolic diseases. And the talk today will focus on liver gene regulation. And want to say thanks so much, Karen, for joining us in the very early hours in North Carolina, and really look forward to your talk.
All right, well, thank you very much. I really appreciate the invitation to present, and I do wish that I were there in person. So as we all know, the pathogenesis of metabolic traits involves many different tissues and many different processes. And some of these we know a fair bit about, some of the processes that influence how free fatty acids are released from the adipose tissue, how glucose is taken up by the various tissues. We learned about some of the specific, genes and proteins, such as PTP1B that we heard about in the previous talk. I'm going to focus today on processes that happen in the liver, and we're going to use a genetic approach to understand what genes we don't yet know about that influence these biological processes. So by taking a genetic approach, I mean we're going to use the natural human variation, the genetic variants that exist in all of us, and relate those genetic variants to metabolic traits and diseases. So this can be done through genome-wide association studies. So in these studies, the genetic variants are tested for association with various traits. So shown here is an example of a genome-wide association study done with plasma alanine aminotransferase levels. So liver enzyme levels that are measured in the plasma. And these can be used as a marker for non-alcoholic fatty liver disease. So shown in this plot on the bottom is the chromosomal position across the genome, and the y-axis is the evidence of association for any given genetic variant, its association with these alanine aminotransferase levels. So every dot represents a genetic variant, and those towers represent sets of genetic variants or loci in the genome that include these variants that influence association. However, the genes that are responsible for these associations are often remain unknown, and we don't know which specific variants affect gene function. And if we can identify the specific variants, then we can use these to test the particular genes to validate the mechanisms by which these effects happen. So if I zoom in on one particular region of the genome, shown here is a little bit more than 100 kilobases of genome region, and every dot, again, is still a genetic variant associated with these alanine aminotransferase levels. The genes are shown below, and you can see that set of variants. There's a lead variant that shows the strongest association, but there are other variants that show very similar evidence of association, and any one of those brightly colored variants could be responsible for this association, and it's difficult to know exactly which gene or genes these variants are regulating. So to understand then these associations between variants and metabolic diseases, we use genomics approaches. So one of those is to evaluate genetic variants that are associated with gene expression levels. So identifying variants that say, may influence the promoter function or enhancer function of a gene, and then using the variants that are associated with both the metabolic traits and gene expression to hypothesize that the variants may act through that particular gene to influence a trait or disease. And then to better understand how those effects may happen, we use the chromatin context. So the genetic variants that are associated with chromatin context can explain the variants associated with expression and then those genes' effect on disease. So going back first to this step of the genetic variants and their association with gene expression and focusing again on the liver, we participated in an analysis of liver, so expression, quantitative, trait, loci, these variants associated with expression levels in the liver. And this is an analysis of around 1,100 individuals for whom the genetic data exists across the genome, and gene expression levels from liver tissue were measured, in this case by microarrays. And so we test the association of the genotypes with gene expression levels to identify eQTLs, expression, quantitative, trait, loci. And in this case, it was 4 different data sets that were combined together, and the analysis identified eQTL from across sets of studies. And in this study, they identified more than 15,000 genes with a significant eQTL. 15,000 genes where at least one genetic variant is influencing the gene expression levels present in liver across individuals. So which of these gene expression levels are related to the clinical traits? An important concept here is the co-localization of these association signals. So the shape of the plot that I showed previously that showed some variants that showed stronger association, others that showed weaker association. If we look in a region and we identify variants associated with some particular trait, we cannot just simply look up those variants in a data set of gene expression levels to determine whether there's evidence of association. We need to determine whether it's the same whole pattern of variants that show that association. So if the same pattern is observed, like on the bottom plot, then we can make that hypothesis that variants associated with the trait and with gene expression levels, that perhaps the variants act through those gene expression levels to influence the trait. So I'll use this co-localization to compare association signals of variants with traits and with gene expression and with chromatin accessibility. So here is an example of comparing the variants that are associated in the top plot with those liver enzyme levels. That's the plot I previously showed you. The bottom plot is these variants associated with the liver expression levels of the gene EFHD1. So the top plot is data from a GWAS study, a genome-wide association study of 162,000 individuals. And the bottom plot is the evidence of association with expression levels in not quite 1,200 individuals. And the patterns of the same lead variants show association in both data sets. So we can use these data to figure out direction of effect. In this case, the alleles associated with higher levels of aminotransferase are associated with higher expression levels of EFHD1. What is EFHD1? It encodes a protein, a mitochondrial calcium binding protein. Other studies have shown that knockout of EFHD1 in neurons alters metabolic stress signaling. So perhaps it also plays a role in metabolic stress in other tissues. So we observe variants that are associated with traits and gene expression. What about chromatin context? So by chromatin context here, I'm talking about chromatin accessibility. So shown in the top plot, the assay we're going to use to analyze this is called a TAC-seq, assay for transposase accessible chromatin. And it's essentially identifying regions where the DNA is unwound. So the DNA shown there is wrapped around histones, except in that place where transcription factors like that dark blue transcription factor are binding to the DNA. So this assay identifies the regions that are accessible to the transposase, which can allow us to isolate the DNA that is in this accessible region, and then we can sequence that DNA, align it back to the genome, identify regions where the DNA piles up in the genome, call these peaks. And these are the regions, you can think of them as regulatory elements that are potentially influencing gene expression. So in this study that I'm going to describe, we used liver biopsies from 20 individuals and performed a TAC-seq and RNA-seq to measure gene expression levels in these individuals to identify the genetic variants across the 20 people that are associated with this accessibility. And this is largely the work of Kevin Curran, formerly a graduate student in the lab. So what do I mean by identifying variants that are associated with chromatin accessibility? So I'm going to call these chromatin accessibility quantitative trait loci, or caQTL. Shown here is an example of a pileup of those reads, or a TAC-seq peak, where homozygous individuals for one allele show strong accessibility, a large peak. Heterozygotes show a moderate peak, and homozygotes for the other allele show a smaller peak. So this is what we're looking for. We're looking across individuals to identify which genetic variants show differences based on genotype across individuals. So in this analysis, we looked at those 20 individuals, looked across the genome in those 20 and identified a little bit more than 3,000 peaks, regulatory elements where there appears to be genetic association. What does this look like? Here are genome browser tracks of the TAC-seq data from 9 different individuals. So as you can see, in the middle of the screen, there are some of these chromatin accessibility regions that look very similar across individuals. Whereas there are others, say over here shown by the purple arrow, that differ between individuals. So I call these CA peaks, chromatin accessible peaks. And in this particular example shown in purple, there's a genetic variant located within the peak where the genotypes are shown on the left. So individuals who have 2 T alleles show the greatest accessibility of chromatin in this region. The heterozygotes show medium level accessibility, and the homozygotes for the C allele show very little accessibility. This is what we're looking for. So to characterize these caQTL, to determine if they really are regulatory elements, we evaluated the fraction of the nucleotides that overlap each chromatin states from the Roadmap Epigenomics Consortium. So the genome's characterized by promoters, enhancers, and other regions. You can see that the regions that are spanned by the chromatin accessible, the peaks that are genetically regulated, the CA peaks shown in purple, are more enriched in promoters and especially in enhancers in liver tissue across the genome. So regulatory elements for liver. One way that genetic variants may affect chromatin accessibility is by disrupting transcription factor binding sites. So we analyze these data of around 4,500 variants that are located within a chromatin accessible peak, a caQTL peak, and that are either the variant itself that is associated with that accessibility or one that's inherited in the same pattern as that. 68% of those altered the binding affinity of a transcription factor. If we think of it from the peak perspective of almost 2,800 CA peaks that contain a variant, more than 80% of those contained at least one variant predicted to disrupt a motif. We further examined these caQTL variants that may disrupt transcription factor binding sites to determine which transcription factors they might disrupt more often than others. So disruption of motifs for 29 transcription factors were associated with whether it was a genetically regulated peak or not. They were associated with chromatin accessible QTL status. And the factors here include members of the H and F family, FOXA transcription factors, CEBP, also CTCF and ATF2. So members of the FOXA and CEBP transcription factor families are known pioneer factors that can bind to inaccessible chromatin and initiate and establish the chromatin accessibility. So these suggest that these transcription factors especially may be ones that are regulating chromatin accessibility in liver tissue. Okay, so great. We have some genetic variants that are influencing these regulatory elements, the CA peaks, but we'd really like to connect those to genes. So how do we connect these regulatory elements to specific genes? So we used 4 approaches to identify which genes these peaks may influence. The first of these is the location of the peak within a promoter region, so its proximity to a transcription start site. So if it's located within 2 kb upstream or 1 kb downstream of a known transcription start site, then we said maybe that regulatory element influences that gene. Second was to use chromatin conformation data or high C data. So if a genetically regulated CA peak was observed in liver tissue to have a high C loop over to a promoter of a given gene, then we said that perhaps that peak can influence that gene. Third is there's a lot of coordinate regulation of regulatory elements across the genome. And so we can look across the 20 individuals and identify a correlation between distal peaks, these enhancer peaks, and promoter peaks. So if we observe a correlation of such peaks, then perhaps that regulatory element of interest is regulating the gene for which there's a promoter. And the fourth is to compare the pattern of variants that are associated with chromatin accessibility to the pattern of variants that are associated with gene expression. So a caQTL and an eQTL that both are that have co-localized association signals. So if that happens, then we say that perhaps that CA peak is regulating that eQTL gene. So when we use these 4 approaches to predict target genes for these regulatory elements, we observe examples for each of them. So here are the numbers of peaks that are connected to the numbers of genes, and then the pairs of those. So you can see that sometimes there are peaks that are correspond to 2 genes. So in transcription start site proximity, maybe that's because there's a bidirectional promoter, and this peak is in the promoter of 2 different genes. The methods do not completely overlap with each other because some of them work better at close distances, like transcription start site proximity. Others work better at longer distances, like high C loops. And we evaluated how many total peaks and gene pairs were present and how many identified by 2 approaches. So of the peaks that were identified, remember there were around 3,000 peaks identified. We identified more than 1,400 peak to gene pairs. And 69 of those, we had a little extra evidence to support because there were 2 approaches that supported the link between that regulatory element and a specific gene. So among one set of those that showed both this co-localized chromatin accessibility QTL and an eQTL. Now we have accessibility and expression level for the same set of variants. We can evaluate how often the element appears to be consistent with binding of a transcription factor activator versus a repressor. So shown on the top, the region that shows accessibility, or the missing histone there, corresponds to higher gene expression, whereas on the bottom, the region of accessibility in allele 1 corresponds to lower gene expression. So we looked across all of the examples of these co-localized signals and found that 77% of the gene to peak pairs appear to show binding of activators. So these might be more considered proper enhancers, whereas around 23%, it looks like perhaps a repressor is binding. So maybe this is more like a repressor. Okay, so now we have our variants that are associated with chromatin accessibility, and we can link many of these to genes. But what about the clinical traits? That's what we're trying to identify, is molecular mechanisms that could be responsible for cardiometabolic traits. So here we analyzed genome-wide association study loci for 19 traits and selected loci that were collected together in the genome-wide association study catalog, and identified more than 100 shared caQTL GWAS signals. In this case, we couldn't quite do the same level of co-localization because our sample size is relatively small, but this 100 potential examples include more than 50 for which we can predict a target gene, and 85 of which we can predict a particular transcription factor that could be binding to a specific genetic variant influencing this regulatory element. Here are the traits that we analyzed and the numbers of caQTL signals that corresponded to genetic variants for genome-wide association studies. And here, liver enzymes is what I've been talking about as my example throughout this so far. The counts here depend quite a bit on how many loci have been identified in the catalog, as well as the role of liver in particular traits. So in some examples, the genetic variants that are associated with a trait and that are also associated with gene expression level are also associated with chromatin accessibility. So here's this same example where I showed the plots on the left previously. And now you can see larger on the right, the genetic variants that are associated with chromatin accessibility. And below the big plot in the track of peaks, you can see the particular region labeled the CA peak. And that's where the purple variant in all three plots, the purple variant is located within that particular peak, within that regulatory element. And it's located within an intron of this EFHD1 gene. So this is co-localization evidence that the same genetic variants are associated with all three traits and potentially are influencing all these traits. Zooming in on this particular region, now shown are tracks from, again, 9 example individuals, and the purple arrow showing that the people with the AA track show very little chromatin accessibility, the heterozygotes show moderate accessibility, and the homozygotes for the T allele show the strongest accessibility. And then shown below, this region in the intron of EFHD1 has been shown by high C data in liver to be connected to a promoter of EFHD1. So suggesting that the variants are acting to regulate EFHD1. So now we have both an eQTL co-localization and high C data to support the target gene being EFHD1 in this region. We then went and tested whether the genetic variant in this region could influence transcriptional activity. So shown here, we did a transcriptional reporter assays. So compared to an empty vector that does not contain any regulatory element, the transcriptional activity for when including this regulatory element shows enhancer activity regardless of allele. And when we include the T allele as opposed to the A allele at one specific variant in this regulatory element, there's more than a 2-fold effect on transcriptional activity. And this is true whether the element, that regulatory element, is included in sort of the forward orientation or the reverse orientation relative to the promoter and the reporter gene. So this shows that this particular variant could be responsible for the effects on transcriptional activity. This regulatory element is been shown to be the site of transcription factor binding in ChIP-seq data. And we evaluated and we predicted that the FOXA2 transcription factor binding site would be altered by the genetic variant. And so shown here is a electrophoretic mobility shift assay that shows in the lower green arrow there that there's a band representing a DNA to protein complex that's found with the T allele, but not the A allele, meaning some protein from hepatocyte HepG2 nuclear extract binds specifically to the T allele and not the A allele. We can compete it away. That's shown in competed away better with the T than the A allele. That's shown in lane 7 versus 8. This sort of is more evidence of allele specific specificity. And when we add antibodies to the FOXA2 protein, we can super shift that band. So that's the upper green arrow, providing evidence that perhaps FOXA2 can bind, at least in vitro, to this particular site. So together across this set of data, we've identified a specific genetic variant and allele. The T allele at this variant is associated with higher chromatin accessibility. It matches the FOXA2 motif better. It shows better binding in vitro assays to the FOXA2 allele. It shows increased transcriptional activity in those reporter assays in the same direction as the association in human liver tissue samples with EFHT1 expression. And these are all associated with higher alanine aminotransferase levels, suggesting a mechanism by which this variant could be responsible for this genome-wide association study signal. Finally, we validated the element, this regulatory element linked gene by knocking down the regulatory element. So we used CRISPR interference in HepG2 cells that are stably expressing a DCAS9, a CRAB domain, a repressor domain. And we showed that when we use a pool of control guide RNAs and compare that to applying a pool of guide RNAs, bringing this CRAB domain to repress the enhancer in this particular region, that the expression levels of the EFHD1 gene show reduced expression. So connecting each piece of the genetic variant, the regulatory element, to this particular gene. So together, I showed you that genetic variants can have very strong effects on chromatin accessibility. And this was a pilot study, and even in this modest sample size of 20 individuals, we identified more than 3,000 regions across the genome where a genetic variant influences regulatory activity. Integrating the caQTL together with transcription factor motifs and target genes can predict these particular functional mechanisms at GWAS loci. And we identified more than 100 where the caQTL was located at a genome-wide association study signal, and 56 of these for which we can predict already a potential target gene. caQTL discovery, like genome-wide association studies, corresponds to sample size. So we now have very preliminary data from a sample size of 138 liver tissue samples, and we identify almost 10 times more caQTL across the genome, opening the doors to identify many more co-localizations with GWAS loci and with eQTLs. And further, the data that I've described are using human liver tissue samples and the whole set of cell types that are present in there. So we're now also doing a pilot study using single nuclear ATAC-seq and RNA-seq so that we can identify which regulatory elements are accessible in which particular cell types within the liver tissue. And then that can be aligned with the expression patterns of different genes in those same cell types. So together, we've identified genetic variants that are related to metabolic traits. There have been hundreds of loci identified for many metabolic traits. We can investigate all of these using gene expression level and have identified target genes in the multiple tissues, and then use chromatin context to identify specific mechanisms by which genetic variants can be affecting gene expression to influence traits. And hopefully through these processes identifies particular target genes that can be used to develop new therapeutics, to better understand biology, and hopefully ultimately to reduce the impact of these diseases. I'd like to thank my many collaborators, members of my lab at UNC Chapel Hill, my other faculty collaborators at UNC, and at many other institutions across the country. Thank you so much.
So thanks so much for the talk, Karen. I think that was brilliant, both in terms of being quite educational, but also in terms of the science, knowing how difficult it is to connect variants to enhancers and the target genes. And now it's time for the questions from the audience. And while people are thinking, maybe I can to start with one. So you had those 4 different ways of connecting the chromatin accessibility peaks to the target genes, and many connections were identified. But I noted that there was not that much overlap between the 4 different methods. So is that because the different methods are finding different types of chromatin accessibility peak gene connections, or because simply there are many false positives and a lot of noise?
Mostly, I would say that we are underpowered to identify the effects of many of these, the connections between variants in genes. So the limited sample size there meant that we did not identify very many caQTL, and so that limited number of caQTL leads to a limited number of caQTL eQTL co-localizations. There's also effects of the distance involved. So the variants that we even tested for relationship to the transcription start sites had to be located within 1 to 2 kb of that transcription start site. When we look at chromatin interaction data, we mostly disregard the variants that are located so close to those start sites, because they'll be sort of chance connections. And so that's primarily focused on variants that are, say, more than 15 kilobases away. I think that as the sample size increases and we have more power to discover more of the relationships, then especially like the correlated peaks, we will identify many more overlaps between those methods and have more confidence in the links from peaks to genes.
Thanks, Karen. No, I had a very similar question as the one that Thomas just asked. I think I'll just hand the microphone back to you.
Hey, Karen, this is Ruth Loos. Very nice talk. I am curious, for the eQTLs, did you also specifically focus on pancreas at the very beginning? Or the liver?
The data that I described was from liver, microarray data sets from liver to get a large enough sample size. I guess at the moment, there are RNA-seq data sets from liver, but they mostly are in the hundreds of samples, and there's not yet, to my knowledge, been a large data set or a meta-analysis data set that was available for us to evaluate. There are other eQTL data sets for these other tissues. So there are a couple large ones for pancreas that have been described that are meta-analyses across, especially islet, RNA-seq data to identify those, the genetic variants associated with expression levels in the islets. Same too with muscle, brain. There are many other tissue sets available. In this case, we just focused on liver because the chromatin accessibility data was from the liver.
Right. And just another question. From talking to proteomics people, they say like the proteome is far more, or the protein levels are far more important for disease than the expression levels. Have you tried to do some colocalization with pQTLs?
Yes. There is not that much proteomics data available yet from the liver. Protein levels are sort of one step further away from the effect of the genetic variant. So they're influenced by many other things, such as the disease state of an individual, other feedback loops that may be present. So they tend to be a little bit weaker and than the more direct molecular effects. So like the variant effect on chromatin accessibility, which maybe reflects on the effect on transcription factor binding, is a very close and direct mechanism. And then gene expression follows from that. So protein is like that next level away. I think that it will be terrific to see the co-localizations with protein levels. I think we might see fewer in part because of then the other changes in protein levels, I'd say that may be like response to disease or traits or other things like that.
And just one last question for me, out of curiosity, how often, if you look genome-wide, is the target gene also the nearest gene to the lead SNP?
Right. Haven't formally done that analysis, but I would say it's consistent with a lot of other studies like this that say maybe it's around 50% of the time the genetic variant that's identified from the studies, like the lead genetic variant, the closest gene has been proposed as a target. I would say it's perhaps similar here. Little bit harder to say because we haven't done this in the sort of more large, well-powered analysis yet.
We have time for one brief question, if there is any. There's one.
This is Sophia also from the Center for Basic Metabolic Research. Thanks also from myself for a wonderful talk. I'm just interested in, like you showed also the link to the metabolic traits, and I'm just wondering how sure can you be that actually the link between this association is actually explained by or maybe also explained by different tissues or effects linked by different tissues?
This is an excellent point. So these associations do not mean causation, and the genetic variants and the genes need to be evaluated for their effects on gene function in the different potential settings, cell types and tissues. There is a lot of genetic variation and effects on gene expression that is shared across cell types. And so it could be that we're observing things in the liver that are also present in other cell types that may reflect actual function in other cell types that are just also observed in liver. So, perhaps these genetic variants are having their effect in neurons and we are observing them in liver. So it helps us, these analyses let us make predictions, but we really need to do the tests of the genes in the relevant cell types to be confident of how they could be influencing the traits.
Okay, thanks again for a great talk, Karen. And I will now hand over to the next chair, Associate Professor Zach Gerhard Heinz, and the next speaker, Professor Sachin Panda.